Good morning. In Chapter 4, we will actually look at microscopy, staining, and classification techniques that we will use during this semester. This particular picture shows Legionella pneumophilia, one of the bacteria we will look at, under the microscope in using one of our techniques we'll talk about later on. In microscopy, size does matter. This slide shows the average chicken egg at about 4.7 centimeters or 47,000 micrometers. The funny looking little u is the Greek letter mu, and this designates micrometers. If you look at this little tiny square enlarged, you will see the different sizes of organisms such as Giardia, Staphylococcus, and orthopox virus. This scale shows the varying sizes of things that we look at. For example, the tick that you can see unaided is a little over 1 millimeter in diameter versus the red blood cells, which are about 7.5 microns or micrometers. E. coli bacteria is about 1 micrometer long. The T-even bacteriophages of the viruses are about a little over 100 nanometers long, and the DNA double helix is about a little over 1 nanometer. This scale actually shows us going down into the picometer scales as well. This table on page 95 shows the differences of metric units of length. For example, there are 10 millimeters in each centimeter. There are 1,000 micrometers in each millimeter, and there's 1,000 nanometers within each micrometer. There are also 1,000 picometers within each nanometer. Think of it. Use your two small millimeter lines. Put 1,000 hash marks between those two small millimeter lines, and you get micrometers. Put 100,000 lines between each of those millimeter lines, and you would have nanometers. A simple microscope has only one lens. For example, here in this replica of Leeuwenhoek's microscope, there is a single lens. You'll notice that I like to have cartoons or other pictures in here, and so I put in this Far Side cartoon of It's a Mammoth. General principles of microscopy. Wavelength of radiation is based on the electromagnetic spectrum. Magnification, which is one of the reasons we use the microscope. Resolution, how well can we distinguish between points. Refraction. Contrast. In this particular figure, we see the electromagnetic spectrum, all the way from gamma rays on one side to radio and television waves on the other side. The visible light spectrum that we see as white light runs from about 400 nanometers to 700 nanometers. What this is talking about is the length between the crests or the troughs in the wave formation of the light or electromagnetic wave. From the one crest to the next crest is one wavelength. So in blue or purple light, there's only 400 nanometers between each crest of each wave, versus red light, which has a longer wavelength at about 700 nanometers. This also means that blue or purple light has a faster frequency of waves, whereas red has a lower frequency of waves. Gamma rays have one of the highest frequencies, while radio and television waves have very low frequencies. The light microscope is when we use any kind of microscope that uses visible light to observe specimens. There are several different types of light microscopy. We use a bright field light microscopy. There's also dark field microscopy, phase contrast, differential interference contrast, fluorescent microscopy, and confocal microscopy. The compound bright field light microscope that we use in the laboratory has all these various parts, which you should already know. But to review, The ocular lenses, of which we use a binocular scope, meaning it has two oculars. The body, the arm, the objective lenses, we have four on our microscopes. The stage, which holds the specimen. The condenser, which focuses the light. The diaphragm that controls the amount of light. The electric illumination source, where the white light comes from. The coarse focusing knob, the fine focusing knob, and the base. Inside the microscope, we have a variety of different lenses and a prism that is used in refraction of the light from the specimen. In a bright field light microscope, the image from the objective lens is magnified again by the ocular lens. Therefore, to get total magnification, you must multiply the objective lens magnification times the ocular lens. On our microscopes, the ocular lenses are always 10x. Our objectives are 4x, 10x, 40x, and 100x. So we have the ability to magnify 40 times, 100 times, 400 times, and 1,000 times magnification using oil. Resolution is the ability of lenses to distinguish between two closely associated points. A microscope with resolving power of 0.4 nanometers, therefore, can distinguish between 2 points that are equal to or greater than 0.4 nanometers in distance from each other, which is a very high resolution. Shorter wavelengths of light provide greater resolution. On your microscopes, you have a blue filter. What the blue filter does is it filters out all of the light except for the blue or violet light waves coming in from the white light illumination source. Why would this be? The limits of resolution depend upon the instrument you are using. For example, the unaided human eye can actually go down to about 200 microns, that's 0.2 millimeters, versus the compound light microscope that can go down to 200 nanometers. When we talk about the electron microscopes, the scanning and transmission microscopes have a 0.4 and a 0.078 nanometer resolution power. The scanning tunneling microscope that we'll mention but not use has a resolution power of 0.01 nanometers. That's 10 picometers. Of these 6 different boxes, which one has the highest resolution? Did you choose this one? The higher the resolution, the better the image. The refractive index is a measure of the light bending ability of a particular medium. The light may bend in air so much that it would miss the small, very high magnification lens of the 100x objective. Therefore, we have to use immersion oil to refract the light, if you will, to keep the light from bending too much so that it misses the high magnification lens. Here's a nice picture of the ability of water to refract light. You not only see the sheath of the grain, but you see all of the other background information in the bubble of water. When light strikes a different medium, it causes the light to refract or bend. If a light wave actually entered this convex lens at a 90-degree angle, it would not bend. The reason is the light will always leave the surface of the convexity at 90 degrees to the surface. Therefore, what will happen is the light will be refracted. We go to a focal point somewhere past the lens, but from this point the light waves don't stop. They will continue on at this point, diverging to actually enlarge the image. So refraction inverts the image and also helps with magnification. This shows without oil, the oil immersion lens would actually miss some of the light waves. When we put the immersion oil on the slide, however, it refracts the light in such a way that all the light waves will enter into the objective lens. Contrast is important in helping determining resolution. Differences in intensity between two objects or between an object and the background is what contrast is. Staining increases the contrast. It gives us the ability to see different structures at better resolution. Use of light in phase can also increase contrast. For example, the top row here, A, C, and E, are specimens used in a phase contrast microscope, whereas B, D, and F are the differential interference phase contrast. Both of these microscopes use light in phase, but you'll notice the cell in A and B, in phase contrast, we see a lot of the surface, while in differential interference contrast, we see more of the internal structures of the cell. Bright field microscopes are normally compound. We have a series of lenses for magnification. The light passes through the specimen into the objective lens. If we're using the 100x objective for 1,000 times magnification, we must use oil to increase the resolution. We normally have one or two ocular lenses. Our scopes are binoculars. The total magnification will always equal magnification of the objective lens times the magnification of the ocular lens, which on our microscope is a 10x lens. In other words, if you took just the ocular lens and could focus close enough on an image, it by itself would magnify the image 10 times. If you have the 40x objective in use, you have to multiply 40 times 10 to get total magnification. In the laboratory, if we ask you what is the magnification, we are talking about total magnification, not the objective lens magnification. Our microscopes also have condenser lenses that refract the light so that it passes through the specimen on the stage. You do not need to memorize these pathways of refraction, but this will help you see how different microscopes work. The bright field illumination microscope we use in the lab shows dark objects visible against a bright background, such as this paramecium. Light is reflected off the specimen and does not enter the objective lens, therefore the object is dark, while light not passing through the object will go into the lens and be bright. Here's a good example. Here's some bacteria under bright field illumination. You'll notice the bacteria, while stained, but they are dark against the light background. Dark field illumination uses an opaque disc under the condenser lens. This opaque disc blocks some of the light waves entering the condenser and the specimen. Light objects, therefore, are visible against a dark background in dark field illumination. Again, here's our paramecium. The light is reflected off the specimen, and this light is what will enter the objective lens, causing the object to be bright and the background to be dark. Here's a good example of a dark field illumination of a close-up of the common flea. Isn't he a cute little guy with his little mustache? Phase contrast microscopy accentuates diffraction or refraction of the light that passes through the specimen, giving us actually good surface images. Here's our paramecium again. In this case, we use the annular diaphragm to allow not the edge of the light or the center of the light, but the midway light to enter into the condenser and to the specimen. Rays in phase mean that the different light waves are hitting at the peaks and troughs together. Rays out of phase means that while one trough is occurring, a crest on another wavelength is occurring. We use these phase plates in these phase microscopes to actually enhance the surface structures of the organism. For example, this shows diatoms, those microscopic algae present in most bodies of water, both salt and fresh. The nice thing about phase contrast is it does give a nice three-dimensionality to our subject. This picture shows the mouth parts of a tick. This actually shows the center, which is the siphon where the female will suck the blood from the victim, and these flanges actually will expand out while the mouth part is in the victim. And when the tick is ready to loosen, she lays these flanges back down and releases. You'll notice that if these flanges were out, this would make the tick actually be stuck. So in removing a tick, you need to make sure that you get some skin that confirms that you've removed the mouthpiece as well. Differential interference contrast microscopy accentuates diffraction of the light that passes through the specimen just like phase contrast, but in this case it uses two beams of light. And you'll notice we have a light and a dark side of our paramecium here. But now you can really see internal structures such as some of the food vacuoles present. This is a differential interference phase contrast microscope photograph of a snowflake. Had to take that one quickly. You'll notice that you again have a light side and a dark side. Fluorescent microscopy uses ultraviolet light as an illumination source, just outside the visible range of light that we can see with our eye. But we also will use fluorescent dyes called fluorochromes that will absorb the UV light and emit visible light that we can see under the microscope. For example, the spirochete has been stained with a fluorochrome subjected to UV light and are visible in the green-yellow spectrum. Here's a fluorescent microscopy of a single human neuron. Don't know which one is the axon exactly, but certainly you can see all the dendrites. Confocal microscopy also uses fluorochrome dyes. This is still our paramecium. Short wavelengths, blue, just at the edge of the 400 nanometer scale, are used to excite the dyes. What you will also see here is not only do we have yellow-green, but we have red spectrum in the visible light from the fluorochrome dyes. The light illuminates each plane in a specimen to produce a nice three-dimensional image, up to about 100 microns deep. This is a confocal section of the organ of Corti of a mouse ear. Again, you can see the reds and greens. Scanning acoustic microscopy, or SAM, uses sound waves instead of light waves. This is basically based on sonar. The sound waves are reflected back from the object. This is used many times to study cells attached to a surface. In this case, you can see our paramecium again. Now what we see is actually structures on the surface of the paramecium at high resolution. Resolution can go up to about 1 micron using a scanning acoustic microscope. This is a scanning acoustic microscope of coal made at 50 megahertz. This image shows it at lower magnification, while this shows the shelves of the carbon atoms at a higher magnification. Sometimes we need to go outside the light microscope to get better resolution. Light microscopes cannot resolve structures closer than about 200 nanometers, and you think, well, isn't that good enough? But if we have viruses that are in the 30 nanometer range, we need to have something else. That's when we turn to the electron microscopes. Electron microscopes uses electrons instead of light. Electrons actually travel in waves just like light. The electron microscopes have much greater resolving power and magnification. The shorter wavelengths of electrons give greater resolution. Electron microscopes can magnify objects up to 10,000 to 150 or so thousand times magnification. We get very detailed views of bacteria, viruses, internal cellular structures, molecules, and even the very large atoms using electron microscopes. There are two types: transmission electron microscopes and scanning electron microscopes. This is a scanning electron microscope of one of my favorite little mites called Sarcoptes scabii. This is what causes scabies in humans and sarcoptic mange in animals. These little mites are actually burrowed down into the epidermis of the skin. You'll notice that one of the clinical signs of scabies is you itch. I wonder why. A transmission electron microscope, or TEM, uses an electronic gun to create an electronic beam. These beams then pass through electromagnetic condenser lenses and are refracted just as light waves would be refracted. They then hit a screen or film where the specimen has been fluorescently dyed, and we then can view those ultra-thin sections of the specimens. However, specimens usually have to be stained with heavy metal salts, such as lead, cadmium, etc., and so this makes this a biohazard. This actually shows the microscope portion of a TEM. The rest of the machine is the electron gun. Actually, this is much bigger than in the picture. But this shows it passing through the different objective electromagnetic lenses. When you see TEM at the bottom of a photomicrograph, this shows a transmission electron microscope. You also will usually have a scale. This is actually a couple of E. coli and a couple of cocci bacteria that are about 1 micron in diameter. You can see very good internal structures of both types of bacteria. This picture shows our paramecium under transmission electron microscopy. You can actually see great detail of the internal structures. This is a picture of what? Anybody know? This is actually the double helix of a human chromosome. The scanning electron microscope has an electron gun that passes the electron beams through electromagnetic lenses and hits the specimen on the surface of our holder. But in this case, what we actually view through the monitor is secondary electrons that bounce off of the specimen to produce the image. Here's our paramecium again. As the name says, scanning electron microscope. Very good for surface structures. We can see the nice cilia all along the paramecium. Here are some Staphylococcus bacteria that are adhered to a surface. Again, very nice for contrast between surface and objects. While the transmission electron microscope has a greater resolution power, the scanning electron microscope has a lower resolution but can still magnify up to around 10,000 times magnification. When you see SEM, it stands for scanning electron microscope. Probe microscopy, as this picture depicts, actually feels the specimen. Probe microscopy can magnify up to 100 million times magnification. However, in using probe microscopy, we destroy the sample. There are two types, the scanning tunneling microscope and the atomic force microscope. The scanning tunneling microscopy uses a metal probe to scan the specimen. The tip will actually move back and forth along the specimen and use electrons to read the surface of the specimen. In this picture, hopefully you can appreciate the double helix of DNA. We can get resolution with a scanning tunneling microscope to 1/100th of an atom. Very high resolution. The atomic force microscope uses a metal and diamond probe that's actually inserted into the surface of the specimen and reads along the specimen just as the scanning does. You'll notice here are the lines that you can see with the scan. This actually shows proteins and their functional assemblies on the DNA at the single molecular level. Staining helps color the microbes with a dye that emphasizes certain structures. A smear is a thin film of a solution of microbes on a slide or a thin section of whatever we're looking at, and a smear is usually fixed to attach the microbes to the slide and kill the microbes at the same time, keeping them as they were in the living preparation. Here's an example of doing a thin smear. You simply roll the cotton tip swab along the slide, thinning out as we go. We then use different dyes depending on what we want to emphasize. For example, here's an unstained human cell. Here's a stained cell. What are the differences? Just remember that staining will kill the cells. So in order to discover things about cell behavior, we cannot stain them. We have to observe the living specimen. Stains usually consist of a positive and negative ion. In a basic dye, the chromophore, the actual colorant, is a cation. In acidic dyes, the chromophore is an anion. So a basic dye would be attracted to what type of charge? And in an acidic dye, what would it be attracted to? Staining the background, the actual glass slide, instead of the cell is called negative staining, and our specimens stand out as ghosts, basically, against the dark background. Simple stains consist usually of either a basic or acidic dye. Differential stains can be used to distinguish between different organisms. We will see 3 of these. You will do one. The gram stain, which determines whether bacteria are gram-positive or gram-negatives. Acid-fast stains, is the bacteria an acid-fast bacteria or a non-acid-fast? And endospore staining, is it an endospore former or not an endospore former? Specialty stains can be used to see particularly difficult structures, such as the negative stain used for capsules, and the flagellar stain in order to see flagellum on bacteria. Simple stain, usually a single basic dye. For example, methylene blue is a simple stain because there is simply a basic dye. A mordant may be used to help hold the stain or coat the specimen to enlarge it. For example, the Gram's iodine is applied after the culture is stained with the primary stain, crystal violet. The iodine acts as a mordant, fixing the primary stain to the cell wall while lending no additional color to the cell. In other words, a mordant itself does not add color, so therefore it is not a stain itself. It simply helps to enhance the staining technique that we're using. These 2 pictures are identical. What's the difference? Differential stains. The Gram stain classifies bacteria into gram-positive or gram-negative bacteria. Why is this difference important? Well, for one thing, gram-positive bacteria tend to be killed by penicillins, cephalosporins, and certain detergents, whereas gram-negative bacteria in general are much more resistant to most antibiotics. Why would this difference be important to me as a clinician? Why is it important I know whether or not my patient has a gram-positive or a gram-negative infection? The Gram stain consists of 4 steps. The primary stain, crystal violet, which is a dark purple stain, the iodine, which is the mordant, an alcohol acetone wash, and a secondary stain, which is a acidic stain known as safranin. In photograph 1, the slide is flooded with crystal violet for 1 minute and then rinsed with deionized water. If we looked at the slide at this point, all the cells, both positive and negative, will stain purple. In photograph 2, the slide has been flooded for iodine for 1 minute. We will actually do 2 minutes in the laboratory because this makes a better stain. Then again, rinsed with deionized water. The result, the iodine acts as a mordant. What's the difference between slide 1 and slide 2? Can you see the more obvious staining effect that the mordant has allowed us to see? Slide 3, the slide is now flooded with a solution of alcohol and acetone. This is for 10 to 30 seconds. I'll show you how to do this in the lab. Literally, you'd go drip, 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 wash with DI water. The result is the smear is decolorized. If we look at the slide at this point, the gram-positive cells remain purple, but the gram-negative cells we can no longer see. What happened? The decolorizer, the alcohol acetone, decolorizes the gram negatives, so they go colorless. In slide 4, we now have applied the safranin, the counterstain for 1 minute, rinsed with DI water, and blotted dry. The result, the gram positives remain purple, while the gram negative cells are pinkish red in color. Yes, this group of bacteria is present on slide 3. It's simply they are gram negative and were decolorized. This table shows the color of both gram positives and gram negatives after each step of the gram stain. So here we have a mixture of bacteria. Who's gram positive and who's gram negative? What about this guy? We'll talk about this one in the lecture. Acid-fast stain shows whether or not a bacteria is an acid-fast bacteria. Acid-fast bacteria have a waxy cell wall that is not decolorized by the acid-alcohol decolorizer. Two major genuses of acid-fast bacteria cause disease in humans: the Mycobacterium species and the Nocardia species. The primary stain in an acid-fast stain is carbofuran, which is an acidic dye, so therefore it's red. We use the acid-alcohol decolorizer and a counterstain, which in this case is methylene blue. Acid-fast bacteria take up the carbofuran stain, do not decolorize, and therefore remain red. Non-acid-fast bacteria, however, lose the carbofuran with the decolorizer and take up the counterstain methylene blue and appear blue under the microscope. This table again shows the color of acid-fast and non-acid-fast bacteria at the end of each step of the acid-fast staining process. This shows Mycobacterium leprae, the causative agent of leprosy in humans. If you looked at this slide, you'd go, oh, gram positive, gram negatives. However, I have to tell you this is an acid-fast stain. Therefore, what we're looking for are the red or reddish bacteria. Those are going to be the acid-fast bacteria. Everyone else that is not red will be non-acid-fast. Endospore staining. The primary stain of endospore staining is malachite green, and usually we apply the malachite green using a heat method. Endospores actually are a non-reproductive form of bacteria that can survive harsh conditions. The malachite green will decolorize out of the cells, but not the spore, using deionized water, and then we counterstain using safranin. Therefore, you wind up with a greenish to clear endospore and a pink safranin stained cell. Here's the endospores, and the pink represents the vegetative cells or the living cells. In this picture, you'll notice that some of the endospores actually no longer are inside the cell. This is simply because the original cell that formed the endospore no longer exists. It has died under the harsh conditions. That's why the bacteria formed the endospores in the first place. Capsular stains, using a negative stain and or a counter stain, is used oftentimes to distinguish capsules around bacteria. Sometimes the cells are stained and then the background is stained with the negative stain. Sometimes we only stain the background. The other specialty stain we'll look at is the flagella stain. This shows a negative staining for capsules. These diplococcus are actually surrounded by a thick capsule around the bacterium. In this case, we stained the background in the light pinkish color and counterstained the bacteria with a basic dye. What this shows is the capsule, which will not take up either stain, appears as a halo around the bacteria. This shows a fungus, a yeast actually, with a very thick capsule. In this case, we only use the negative stain. It's known as an India ink stain. And this shows this very thick capsule around this little yeast body. The flagellar stain, we use a mordant, usually iodine, with this staining process. This will help to emphasize the flagella on the bacteria. Carbofuscin is the simple stain that we use to stain the cells. This shows the flagella staining of Proteus vulgaris, one of our common bacteria. Can you see all of the flagella? In classifying and identifying microorganisms, we use taxonomy and identifying characteristics. We've discussed these, but physical characteristics, what do they look like? Biochemical testing that we will be doing in the lab, serological testing, phage typing, and analysis of nucleic acids. Two biochemical tests that are used for identifying bacteria are shown here. In this, we are looking for fermentation of a sugar by the bacteria and whether or not it produces gas. In this photo, we are looking for whether or not the bacteria produce H2S, hydrogen sulfide, within the media. We will be doing several different tests in the lab. In Lab 5, we will be doing the tests on the left of the slide. In Lab 6, we will be doing the ones on the right hand of the slide. The OF glucose test, or the oxidation fermentation of glucose, oxidase test, nitrate test, starch digestion, carbohydrate fermentation, the MRVP, which is actually two different tests, the methyl red and the Voges prosaccharide, good thing we call it VP test, and the triple sugar iron or TSI test. In Lab 6, we will look at protein metabolism, the urea test, gelatin metabolism, phenylalanine test, and peptone or indole test all look at protein digestion. Lipid metabolism looks at fat digestion. Bile echelon auger looks at the use of bile. Citrate test, can citrate be used as a carbon source by the bacteria? And motility test, do the bacteria have the ability to move? We will go through each one of these. These are also repeated in Labs 5 and 6 in the laboratory information. The OF glucose test, or oxidative fermentation test. This uses a tube that has glucose in the media. If the bacteria can break down glucose, it will do it in one of two ways, either aerobically using oxygen or anaerobically without the presence of oxygen. You will inoculate your tubes with the bacteria. You will have a set, so one bacteria goes in two different of the tubes. You then will cover one of those two tubes with mineral oil. This sets up an anaerobic environment. OF glucose normally starts out as this greenish color. So, for example, if you have an open tube, no oil, and an oil tube, and both of them remain green, this means the bacteria cannot ferment or oxidize glucose. So it's negative for both fermentation and oxidation. However, if the open tube turns yellow, as indicated by the pH indicator that glucose has been utilized, then it's positive for oxidation. If, however, the oil tube remains green, this means this bacteria can only use glucose in an aerobic environment, so it's positive for oxidation. If both tubes, both the open and oil tube, turn yellow, it is positive for both fermentation and oxidation. If the oil tube turned yellow, but the open tube remained green, this would mean the bacteria is only a fermenter of glucose. The oxidase test shows whether or not the bacteria can use the oxidase enzyme. Positive on the oxidase test will be a purple color, negative will be a clear or brownish color. The nitrate test. We start out with nitrite in our nitrate media. We inoculate the bacterium, and then after incubation we'll add nitrate agent A and nitrate agent B. Nitrate agent A is sulfonolic acid, while reagent B is alpha naphthylamine acid. When nitrate agent A and nitrate agent B are added, if the tube turns red, this shows that we have broken the nitrite, NO3, down to NO2, and therefore is considered positive. If there is no change, however, after adding nitrate A and B, then we must add zinc dust to the tube. If the tube remains a yellowish color, this means that we have a positive reaction. However, if it's still yellow after the addition of nitrate A and nitrate B, and we add the zinc dust, and the tube turns red now, the test is actually negative for nitrate use. Here is an uninoculated nitrate test. To tube number 1, nitrate A and B have both been added. Therefore, at this point, this is an inconclusive test. It stays the yellowish clear color. We've added nitrate A and B to tube 2. It turned red, therefore it is a positive test, and we are now done. However, if you have tube number 1, you then must add zinc. If tube number 1, after adding zinc, remains yellow, as in tube number 3, the nitrate test is a positive test, but if at this point the tube turns red after nitrate A and B and zinc have been added, as in tube 4, this is a negative test. Starch digestion, we will actually put bacteria on a plate of starch agar. After we do the oxidase test, we will add iodine to the plate. If a clear halo is seen around the bacterial culture, it is positive for alpha amylase production, or it means it can digest the starch, and this would be a positive starch test. Carbohydrate fermentation, we can use different sugars in the liquid medias to determine what type of sugars the bacteria can ferment. In our testing, we will utilize over the semester glucose, fructose, mannitol, sucrose, lactose, and maltose sugars. All of these are described in the carbohydrate fermentation test in Lab 5. One of the things you'll notice with these carbohydrate fermentation tubes is that there is a smaller tube actually upside down in the test tube in the media. This is known as the Durham tube. This will test for whether or not the bacteria produces gas during fermentation of the sugar. This tube shows an uninoculated tube. They start out reddish in color. If no sugar is fermented, it remains red. So this would be a negative test. However, if the sugar is fermented by the bacteria, it will cause acids to be produced and cause a change in color to yellow. This tube, therefore, is positive for carbohydrate fermentation. In the third tube, you'll notice that there is a gas bubble inside the Durham tube. During the fermentation of this carbohydrate, this bacteria also produce some form of gas, some of which is trapped in the Durham tube. We would then label this one as positive/gas for a positive fermentation and gas production. The MRVP test is two different tests using the same original media. It is what you add after incubation that will determine whether you're doing an MR test or a VP test. For an MR test, you will add the MR reagent after incubation of the bacteria. The uninoculated media is a yellowish color. If after adding the MR reagent and mixing, the reagent turns red, bright red, it's a positive MR test, as this middle tube shows. However, after adding the MR reagent, if it remains yellow or turns slightly orange, it is a negative MR test. The VP test starts again with the same media, but you will add VP1 reagent and VP2 reagent after incubation in order to read. If, upon adding VP1 and VP2 reagent, the test media turns red, it's a positive test for VP. If it remains yellow or does not turn red, it's a negative VP test. The MR test checks for the presence of alcohol made by the bacteria. The VP test tests for the presence of acids. The triple sugar iron test has 3 sugars in the media. In the slant portion of the tube, we will have sucrose and lactose. In the butt or bottom of the tube, we will have glucose. TSI results are reported as the slant result / butt result / H2S if present / gas if present. For example, a tube may be negative on the slant, positive on the butt, and have H2S in the butt. Or it could be positive on the slant, positive on the butt, have H2S in the butt, and have produced gas bubbles. Or both the slant and the butt could remain negative. Or you could have a negative, positive gas. The slant being negative, the butt being positive, but you have gas bubbles. If the butt is all black, the butt reaction is still positive. How could you get H2S if no glucose is fermented? We'll discuss this in lecture. Here are some examples of TSI. This is the control. It's reddish to begin with. Upon inoculation, if neither the slant nor the butt turns yellow, this is a negative / negative result. In this tube, the slant is red, but the butt turned yellow. This would be reported as negative / positive. This tube shows mostly a yellow slant, all yellow butt, but this is a gas bubble produced within the media. So this would be labeled as positive / positive / gas. This tube shows a yellow slant, an actual yellow butt, with hydrogen sulfide produced. This would be positive / positive / H2S. This tube shows a red slant, yellow butt with hydrogen sulfide. This would be reported as negative / positive / H2S. Protein metabolism in Lab 6. These following tests are used for protein metabolism: urea, gelatin, phenylalanine, and the peptone indole test. The urea test we buy commercially. The uninoculated tube is in the center. It's kind of a peachy yellow color. If the test tube turns hot pink, it is positive. Any part of the test tube being hot pink is positive. The negative tube is on the right. This shows no color change or a slightly color change to yellow from the original uninoculated tube. The gelatin test is a very thick gelatin that you inoculate your bacteria into. You then will incubate, and out of the incubator, both tubes, positive and negative, will be liquid. Therefore, they have to cool in an ice bath in order to read. If the tube remains liquid after having time in the ice bath, it means it had the gelatinase enzymes, therefore is positive for gelatin digestion. However, if the media becomes solid, it is a negative test for gelatin. The phenylalanine test must have ferric chloride added after incubation. If the slant remains yellowish in color after the addition of the ferric chloride, it is negative, as this test tube on the left. If the slant turns greenish after about a minute, it's a positive test, as this one on the right. The peptone indole test must have Kovacs reagent added after incubation. This one you do not want to mix. You simply want to see, is there a cherry red ring on the top of the media? If it is, as in this test tube on the left, it's a positive peptone indole test. If it's yellow, it's a negative peptone indole test. A lipid metabolism test that we will use is known as the spirit blue agar. This has had a lipid, actually milk lipid, added into the media. A lipase positive spirit blue organism will have a blue halo around it, as the Staphylococcus epidermidis shows. The E. coli actually clears the bluish color and therefore is lipase negative. The bile echelon agar or bile agar is a slightly brownish greenish color prior to inoculation. If it remains this brownish yellowish color, then it's a negative test. However, if 50% more of the bile actually turns black, it is considered positive for bile metabolism. The citrate tests test whether or not citrate can be used as a carbon source by the bacteria. It starts out as this teal green color here on the left, and if the bacteria can use citrate as a sole carbon source, it turns a brilliant blue color. Again, any blue color in the media is considered a positive citrate test. The motility test will be inoculated with your inoculating needle. You make a straight stab line down the media. We are not looking for a color change here, but we're looking for where growth occurs. If the line is a sharp line, as in test tube B, the bacteria grew only where the needle was put, then the bacteria is not motile. However, if you have a fuzzy line or no line within the media, as shown here in A, then you are positive for motility. This would mean most likely the bacteria has flagella. Serologic testing that we can use, one of these would be an agglutination test. Probably most of you have done a plate similar to this with blood typing. What we are looking for in an agglutination test is the interaction of antibodies, these structures, with the antigens present on whatever cell we're looking at. So for example, if you put the anti-A antibody in with the patient's blood and you see no agglutination, it means that the A antigen is not present. But in this case, in the B well, we put the anti-B antibody in and it clumped the cells up, agglutination, because the anti-B antibody interacted with the B antigen on the surface of the red cells, as would be shown in this drawing. The same thing happened with the RH well. So we are looking for agglutination. If the correct antigen is not present when we add the specific antibody, there will be no interaction, therefore no agglutination is shown in this drawing. Phage typing is a specialized test that we will not be utilizing in the laboratory, but we use actually different bacteriophages that interact with different strains of bacteria. This plate actually shows a lawn of E. coli. However, you'll notice that different areas or plaques have different appearances. This is because different bacteriophages are acting and killing different strains of the E. coli within this E. coli population. Therefore, we have different plaque appearances. Taxonomic keys. We will be using a dichotomous key in order to identify the unknown bacteria. This is a series of paired statements where only one or two either or, yes, no, positive, negative choices applies to any particular organism. The key directs the user to another pair of statements or provides the name of a specific organism. For example, if I were to go into our prep room and make a dichotomous key of all the scientific glassware present, I could separate them into two categories, round bottom glassware and flat bottom glassware. I want to put them in particular cabinets so that everybody would know where the round bottoms and flat bottom lab glassware is located. I could then separate both round bottom and flat bottom into glass and plastics. So if I had a flat bottomed glass flask, I could separate those into wide openings and small openings. So the flat bottom, glass, wide opening glassware will be put in cabinet A, whereas the flat bottom, glass, small opening will be put into cabinet B. If I have a flat bottom plastic glassware, I would put it into cabinet C. I am now done with all of the flat bottom glassware. I then turn my attention to the round bottom glassware. Again, separating the round bottom glassware into glass and plastic. The round bottom plastic I'll put in cabinet F, but if it's a round bottom glass with a wide bottom, I'll put it in cabinet D, but if it has a small bottom, I'll put it in cabinet E. So if someone comes in and needs a round bottom glass, small bottom glassware, all they'll have to do is go scientific glassware, round bottom, glass, small, and go to cabinet E to find what they are looking for. We use many different things in order to build our dichotomous keys. We'll use gram staining. Are they gram positive or gram negative? We'll use the three shapes: coccus, which is round, bacillus, rod-shaped, or spiral organisms. For your unknown in lab, we will only be looking at cocci or bacilli. We then look at the special biochemical tests in order to separate out the bacteria. This shows a dichotomous key in two different forms: the flowchart form on the right and the statement form on the left. I prefer that you use the flowchart for this class. So, the first question, gram-positive cell, yes or no? In this case, we have a gram-negative, so it's a no on the gram-positive. Are they rod-shaped, yes or no? In this case, we have one that is rod-shaped, yes. Can this gram-positive rod-shaped cell tolerate oxygen? The answer in this case is yes. Now the next question would be, can this gram-positive rod-shaped cell that tolerates oxygen ferment lactose? Yes. Can then this gram-positive rod-shaped cell that tolerates oxygen and ferments lactose use citric acid as a sole carbon source? And in our case, the answer is no. Then, can this gram-negative rod-shaped cell that tolerates oxygen, ferments lactose, that cannot use citric acid as a sole carbon source, produce gas from glucose? And the answer is yes. Therefore, I have now identified my bacteria as Escherichia coli. The same key is located in the statement form. I get lost in this, but for example, you can say 1A and B, gram positive or gram negative. We said it was a gram negative, so we go to statement number 2. Is it rod-shaped or not? It's a rod-shaped cell, so we go to statement number 3. Can it tolerate oxygen or not? It can, so we go to statement number 4. Can it ferment lactose or not? It can ferment lactose, so we go to statement number 5. Can citric acid be used as a sole carbon source, or can it not be used alone? We said that we cannot use citric acid as a carbon source, and so now we have to go to statement number 8. Does it produce gas from glucose or does it not? If it produces gas, then it's echerichia. I get lost many times in trying to figure out which statement I'm going to. Therefore, when you build your dichotomous key from all of the unknown bacteria that I will give you later in the semester, you will do your flowchart like this. So here I have 8 different bacteria. I'm going to separate them into 2 groups, the gram-positives and the gram-negatives. I first look at my gram-positive bacteria. Then I have to separate into who is a coccus and who is a bacillus. Enterococcus faecalis and Staphylococcus epidermidis are gram-positive cocci. Bacillus cereus and Bacillus subtilis are gram-positive bacilli. I then want to look at my gram-positive cocci. What would separate my gram-positive cocci from each other? And I see that a bile test would separate them because Enterococcus faecalis is bile positive, while Staphylococcus epidermidis is bile negative. The same thing I would do with the gram-positive bacilli. I would look for a test that would separate Bacillus cereus and Bacillus subtilis. And MR does this nicely because Bacillus cereus is MR positive, whereas Bacillus subtilis is MR negative. Now I am done with all of my gram-positives. I don't have to look at them anymore. I now turn my attention to the gram-negatives, of which I have 4. I will separate these into gram-negative cocci and gram-negative bacilli. The 2 gram-negative cocci, Neisseria sicca and Neisseria subflava, can be further separated into individuals by the lactose test, whereas Neisseria subflava is lactose positive and Neisseria sicca is lactose negative. Now I turn my attention to the gram-negative bacilli, Enterobacter aerogenes and Pseudomonas aeruginosa. I can see that a mannitol test will separate these 2 because Enterobacter aerogenes will ferment mannitol with gas production, but Pseudomonas aeruginosa is mannitol negative. So, if I were to ask you what is a gram-negative bacillus that ferments mannitol and produces gas is... What about a gram-positive bacillus that is MR positive? What is the name of this organism? We will do many of these during lecture.